Are just on time, the session number 4, which is going to be chaired by Maria Saponari from CNR Italy. Thank you, Maria.
So we are ready to start the last session for today. During this session, the speech are related to some tools and application for controlling the bacterium in the plants or attempts to mitigate the effect of the infection, and some new and novel research we started here in order to hope to develop some tools for the containment of the bacterium. So the first speech will be given by Alessandra de Souza. Alessandra comes from Centro di Citricultura Silvio Moreira in Cordeiropolis in São Paulo, Brazil, and she is leading a research group working on pathogen of citrus and mainly on Xylella fastidiosa, with some, the majority of her activity is on the working on resistance on Xylella fastidiosa and other tools for the control of the bacterium, and she will introduce this section. Some problems.
Should there? No, I know that it's blocked the computer. It's not working.
Quel programma.
I'm so sorry. I don't know. It's not fun. It's blocked.
If you want to try my pen drive.
But this is a problem for the power.
You have it on the pen drive.
It's blocked and I can put presentation because it's blocked.
What blocked? Enable content? Try to enable. It doesn't go. My presentation was there. Yeah, this was what I was working on. The runs the computer. Try to control out del to see.
It's blocked.
Yes, completely.
Con i luci.
No?
She tested before you worked in the computer.
We must reset. I think reset.
It's not needed. It's not needed. Yes.
Start again.
Yeah.
I still have 25 minutes.
Try to replace it with this one. Ah, you're starting. It's the computer that is blocked. Ah, okay. Because these are very heavy presentations.
No problem.
Yeah. So many in front of you. Yeah. Everybody looking for us. Yeah.
I think it was
A little bit campus, running in the streaming lights.
Like press conference.
People in my lab, they are looking for.
Imagine, I have such a piece of experience like you. So
I have different experiences.
Yeah.
This is they are expert.
Yeah, true.
Even students,
Yeah.
they are rowers or.
Yeah.
But here are real knowledge. So question.
Dame los cables.
Okay, we can start.
I want a pointer. Okay, good afternoon, everybody. I still have 25 minutes. Huh? And so, first of all, I'd like to thank the organizing committee for the invitation and also for the opportunity to show you part of the results. We are working on the Citrus Research Center that belongs to Agronomic Institute of Campinas that is in São Paulo State in Brazil. And so the results I'm going to show is related to the advances in controlling Xylella fastidiosa in host plants. Actually, it's more specific to citrus, but it could be applied to other hosts, other plants, right? And so why citrus? Very quickly, because this citrus is a very important crop for our country, since we are responsible for approximately 60% of all orange juice produced in the world, which brings approximately $1.5 billion a year to Brazilian agribusiness. However, even though this number is good, so we are surviving, okay, but the problem is that the economic gain of the citrus grower has decreasing, and it because the significant increase of phytosanitary problems that affect the citrus. And so in this slide is only some example of disease that affect the scion part of the plant and also disease that affect the rootstock. Nowadays, the main disease that is affecting the citrus production is HLB in Brazil and also in the US. But 20 years ago, the main disease was CVC. It's 20 years ago. So very quickly, I'm going to show the timeline of citrus variegated chlorose, that is CVC in São Paulo State. It occurred in 1987, it was the first description, and the Koch postulate was in 1992. In 1995, Dr. Purcell helped us to identify the sharpshooter that was transmitted, Xylella fastidiosa. And in 1997, we had the worst situation, the bad situation, and where 31% of all orchards had the incidence of the disease and 3% in the terminal phase. And in this situation, the government started to put money in the research, and one of these research was the sequence of the genomes of Xylella fastidiosa. Nowadays, to sequence a genome is very easy, but it was not in that time. So it was the first phytopathogen to be sequenced, that the genome was sequenced in the world, and so it was the cover of the Nature. And after that, increased the number of the scientific research and paper related to Xylella fastidiosa and citrus. And so, and now, and what now? After all this investment and research in relation to Xylella, now we have this situation that is very nice. So we decreased the situation one that is approximately 50% in 2008 to now we have only 3% of the CVC incidence of the disease in São Paulo State. And why? What's the practice we are doing to get these results? We have three practices that we are using that is very important. One of them is the healthy seedling produced in greenhouse to prevent the Xylella transmission by the vector. The other one is pruning and eradication of the disease in plants. And the third one is the chemical control of the vector. Very quickly, so we have a legislation that was created in 2003 where all seedlings must be produced in protected nursery. The second one is the pruning. So when you have a situation like that, so you have symptoms in the leaves, but you don't have symptoms in the fruit. So in that case, it's possible to pruning and keep the plant productive. But when you have this situation, a severe symptoms in the leaf, the fruits are already small. So in that case, you have to eradicate the plant. So the situation like that. And then the third one is the chemical control of the vector. We have at least 30 different species of the sharpshooter vector of Xylella fastidiosa. And so we have to control using insecticide. So in general, the incidence has fallen year after year in the younger orchards that adopted the proper management of the disease. However, in relation to this one, these have the increase, the use of the insecticide, the huge amount of the insecticide. Even though it's necessary, it increased the cost of the production, and so as a consequence, decreased the economic gain of the citrus growers, and they also increased the environmental impact. There is another problem, that is the climate change. Why? Because we have a risk for CVC increase in São Paulo state, because the main incidence of the disease occur in a region that with high temperature and hydric and water stress. And this kind of climate became more common in the São Paulo state because we have now a period of water stress. It's increasing in the state. We have a period of drought that is increasing. So there is a risk that that region, that the incidence is low, become that increaser again. So what we are doing, what we are working in a more sustainable methods to try to control CVC. And so in my institution, we are working in three main approaches, that is breeding, transgenic plants, and the use of this molecule N-acetylcysteine. I'm going to talk very briefly about the breeding we are doing there, transgenic plants, and so the majority of my talk will be regarding the use of this molecule. In relation to breeding, so we have a very big program in our institution that Dr. Mariana Christophe is the responsible for this research. She is here in the audience, so if someone has some question about this, she can help. And so the idea was, it occurred 20 years ago, was crossing the mandarin. Actually, this kind of mandarin was a cross already, natural cross between Citrus sinensis, that is susceptible to Xylella fastidiosa, and the Citrus reticulata, that is resistant. And so it is kind of back cross. And so she got 264 hybrids from this crossing. And she tested this with high CVC pressure. And so this hybrid was challenged with Xylella using a bud from a symptomatic plant, and she grafted this bud in all the hybrids. It's to make sure that all the hybrids was infected with Xylella. And so after that, she could verify that all the grafting, the bud, showing CVC symptoms, and she evaluated the rest of the hybrids in relation to Xylella pressure. And also she evaluated by PCR to make sure that all hybrid was infected in the beginning. And so we have a experimental network in São Paulo state with different hybrids in different locations that produce citrus. And very quickly, she obtained 8 new hybrids that have the sweet orange phenotype, but are resistant or tolerant to Xylella. And so these hybrids are already distributed to growers for field trials that is related to orange juice quality and also production, productivity, and the other characteristics. And now she's working on gene expression. So what gene expression occur in this resistant variety that could explain the resistance to Xylella. And she has a poster that will be present here, so you are invited to see the results of this gene expression associated with this hybrid resistance. And so the next approach is regarding the transgenic plants. So we are working on two different approaches. One of them is using genes from mandarin because it's Citrus reticulata. So the idea is to get genes from citrus to citrus in the approach that we named seed genic because it's citrus from citrus that could occur naturally, but with transgenic we are get more faster these hybrids. And the other one, not hybrids, I'm sorry, faster the sweet orange carried resistant genes from mandarin. And so you can keep the sweet orange characteristic, but carrying the resistant genes. So we are working on different genes in this approach. And this one is, we are working on take the genes from Xylella fastidiosa and transform it sweet orange. We are working the RPFF gene that is responsible for DSF, the production of DSF. This idea came from Dr. Steve Lindau in California. He used these genes to transform grape, and these plants are now in the field, and it's showing good results. And we have a collaboration with him, and we did similar in citrus. And recently, actually this month, we published this paper where we show that citrus expressing these genes are more resistant to CBC. And we're very happy because this work is the cover of the MPMI journal of this month, where here we show two different citrus varieties that is non-transgenic, showing severe symptoms of CBC, and the other part is the transgenic plants showing much less symptoms. And then, right now, we are working on this other approach that is transform the rootstock with these genes, because the idea is if the rootstock could translocate it, the transgenic rootstock translocated the DSF from the root to the xylem, maybe the plant will be more resistant to Xylella, but the fruit will be not transgenic. So this work is going on. And so the third one, that's the use of N-acetylcysteine to control Xylella fastidiosa. And so where did this idea come from? I have to tell you the story very quickly. And so everybody knows already that was shown here that biofilm formation is very important to the symptoms of CVC. So, and we verified that biofilm formation involves the expression of the ephemeral and ephemeral protein, because it's important to biofilm formation and also to the movement of the bacteria in the xylem. And so these proteins are expressed in the Xylella in different hosts, citrus, periwinkle, and hibiscus. We showed that in 2010, I think. But what's very interesting is because the mechanism of the bacteria, the protein interaction of Xylella's cell-cell interaction is quite similar what the protein used by bacteria that cause human pathogen to interact with each other and form a biofilm. And so the interaction of these proteins it's depend of the disulfide bonds, okay, to occur, the adhesion and also the folding and the stability of the proteins. And there is this model was already showed by De La Fuente today, and Xylella fastidiosa, it was showed in 2002 that Xylella fastidiosa use, besides calcium, disulfide bonds to adhere each other. And as Xylella fastidiosa has the mechanism for adhesion quite similar with the bacteria that cause human disease, we thought which is used in this bacteria that could be used also in a plant pathogen. And so we found this molecule, N-acetylcysteine, and the N-acetylcysteine is used in the human, especially to control the respiratory disease because it breaks disulfide bonds and this interaction in the mucus too. And so what is this? What is N-acetylcysteine? N-acetylcysteine is a cysteine analog. So, and some properties that we can find in the literature, in some paper, is that NAC has been attributed to decreased biofilm formation, which reduces bacterial infection, and it has antibacterial properties. And we find in literature a lot of papers, you can find there are a lot, showing that NEC could control bacteria that cause human disease, gram-positive bacteria, and also gram-negative bacteria. But at that time, there is no paper showing that the use of this molecule to control a plant pathogen bacteria. So we published this paper in 2013, where it showed that, yes, NEC can be used to control Xylella fastidiosa. We showed that this molecule disrupts the biofilm in vitro, and also in plants. So we observed that in vitro we got very nice results, but the question was, and in plant, because in human can take the medicine, but how can apply this in plant? And so also quickly we test using fertirrigation treatment and fertirrigation plus injection to see if it will be better. And we observed here that injection doesn't work, so it could be by fertirrigation. If you observed here, before NEC treatment, this is without NEC, in the initial symptoms of CVC, 8 months after NEC, we can see the increase of the symptoms. But with NEC and initial symptoms of CVC, we could observe a remission of the symptoms. And here with NEC and with severe CVC symptoms, there is no remission, but the severity of the disease is decreasing. And so, but when we stop the treatment, what occurs is that 3 months after, the symptoms come back. And why? We analyzed the NEC degradation, and we observed that in solution, after 15 days, the NEC is completely degraded. And so we thought we have to improve the way of NEC application to get a slow release, because we believe that this NEC is more slow released to the plant. The plant will have more time to absorb the NEC, and you'll get better results. And so it was what we did very fast too. We absorbed NEC to organic fertilizer, knowing to be slow released. And so we applied in the plant that has the same level of CVC symptoms. And we observed that the decrease of the severity of the disease do not evolve when the NEC treatment, as you can see with NEC and without NEC. And so, and in that case, the symptoms comes back after 6 or 8 months when we stop the NEC treatment. And so, after that, it was all the results are published already, and after that, we started work on different approaches, like the absorption of NEC by roots and by leaves, the colonization in the plants. What occurred was the flow of the nutrient in the water when the NEC is in the plant, treated in the plant, affecting the site acquisition. But what I'm going to show you today is the effect in field condition, because it's more important. And so, to do that was very difficult, because it's very hard. When you work in the greenhouse, it's one thing, but when you work in the field, it's completely different. There's a lot of plant, people, and whatever. But we got the collaboration of the citrus succo. No, it's not here. I forgot to put the collaboration. I think that's here. Yeah, sorry. The citrus succo industry helped us, and they got the field. This field was 30 years old, showing severe CVC symptoms. There's no irrigated ocean. And so here it's important, because severe CVC symptoms, it means that the fruit was already small. It's so severe. And so we use a different kind of NEC application. And so I'm going to show only the second year of the results we got. And here is an overview of the field experiment by soil. We also spray the NEC, and also by trunk, and the NEC absorbed in the organic fertilizer was actually also at the NPK. And so what we evaluated in the second year was the fruit diameter, production, kilogram the fruit by plant, juice yield, and sugar content. And we analyzed the size, the diameter of the fruit before and after the treatment. And then the results. And so in relation to fruit diameter, so that part, it's only in relation to plant showing CVC symptoms. And here is health plant, because it's important to evaluate the effect of the NEC in health plant to see if the NEC will be toxic or not. But was very curious. Okay, in that parts, the only treatment that improved the fruit diameter was the NEC adsorbate organic fertilizer that is low release. Spray did not work, trunk also did not work. Only this treatment showed better results. And what was very curious here is because the fruit diameter also increased in the health plant treated with NEC and in the control. Health planted treated with fertilizer without NEC also increased the fruit diameter. But the more important to the citrus grower is the productivity. And about the productivity, okay, the citrus production by kilogram of fruit by plants, because it's the way that the citrus growers sell the fruit. And so in that case, we did not observe a significant results, but in that case, only the treatment of CVC treated with NEC increased 14 kilograms of fruit by plant. But what was more interesting was in that case, because the health plant treated with NEC almost was the double of the production in relation to the treatment without NEC. So a significant increase in fruit production in health plant. Okay, I read that. In relation to fruit production, the juice yield was the similar results of the fruit production, and we did not observe any difference in sugar content or other characteristic of the juice, the orange juice. That is important for the industry to know that. So general conclusion, the test of NEC was done in plants showing severe symptoms of CVC, where the indication is to eradication plants showing such symptoms. Indeed, as already observed in greenhouse experiment, the NEC application in field conditions do not completely recover plants showing severe symptoms. However, as also observed in greenhouse, the NEC application via soil adsorbed in organic fertilizer decreased the disease severity in field, since we observed again in fruit diameter and fruit production. So CVC treated with NEC produced 14 kilograms more than CVC without the treatment. So we observed a partial recovery, and it will be a farmer decision based on the cost benefits if these 14 kilograms will be get more money in relation to NEC application or not. So it's a cost benefit. And we recommend that NEC application plant, we think that NEC application in plants with initial disease symptoms could prevent disease progression and avoid symptoms in fruits. And we recommend more than one application, 2 or 3, because we use it just once, one application in February. And so we observe it in the end of when we harvest. And in relation to NEC in houseplant, the result is that the results we observed that NEC provides other benefits to the plant than not only to eliminate the pathogen, because we observed an increase in production when the health plant was treated with NEC. And besides, NEC disrupted disulfide bond. NEC is also known to be an antioxidant molecule. And so it is used in medicine as a supplement to decrease the oxidative stress in humans. So we thought NEC can also decrease ROS in plants. And so we analyzed the hydrogen peroxide, because we know that it's very toxic to the plant and increase when the oxidative stress is occurring. And we observed that plant treated with NEC decreased the level of the hydrogen peroxide, and the other hand, plant that was not treated increased it. And so the other one is catalase, and catalase is a good enzyme because occurred the detoxification of hydrogen peroxide, and as expected, the increase in NEC treatment. And the lipid peroxidation that is associated with sclerosis and necrosis, and we analyze this enzyme, which is a key enzyme to analyze the lipid peroxidation, and it was significantly less in plant treated with NEC. In that case, we believe that the increase of fruit production on houseplant treated with NEC could be due a decrease of oxidative stress and consequently the improvement of plant performance. And so the conclusion is that NEC application in houseplant could help to avoid plant infection, pathogen infection. NEC application plants with initial disease symptoms could prevent disease progression and avoid symptoms in fruit. And the advantage to use in agriculture is because it's a small molecule, easy adsorbed, and can be combined with other molecules to enhance its action. We think that we have a lot to do to improve this molecule in the plant. It suffers environmental degradation, more sustainable for the environment. So when we take all these results, it was last year, the next question was how to apply NEC in the field. I don't know here in Europe, but in Brazil there is a big gap between the university and our research institution. When you have something, there is a big gap to apply, definitely apply to the consumer, to the farmers. It was very hard to transform NEC in the product, to be finally go to the field. And so last year, my ex-postdoc, Simone, she opened a small company. The name is CIACAMP, that means from the science to the field. And she applied to a FAPESP, that is an institution that supports projects in São Paulo state. And in this kind of program, PIP, that's innovative research in small companies. And the idea is to try to transfer this technology. And so now all the new experiments that will be done, done in the field, will be now using the support of the FAPESP and CIACAMP, and we hope that we get more results to show in relation when the plant is in the initial symptom of CDC, more results in the health plant, and try to see something new to apply in the field condition to control Xylella and probably other phytopathogens. So this is the team. Thank you very much for your attention and invitation.
Thank you, Alessandra. And the next presentation will be by Crescenza Dongiovanni. She works at Centro di Ricerca Sperimentazione Basile Caramia in Locorotondo. She is conducting several field trials for the containment of the disease, but also for the control of the vector. So a research center fill a little bit the gap that Alessandra was saying between us, the group of the university, and the farmers. I take the opportunity to thank Alessandra for the suggestion and advices. She's following some of our experiments, so bringing her background both for the resistance, the study of the resistance in olives, but also, as you will see now in the presentation, for the NAC application for the olive disease.
Allora, good afternoon to everybody. As said Maria, I've tested different products to evaluate the efficacy of different products against Xylella. Since the introduction of Xylella fastidiosa in Apulia and the rapid spreading of the disease, a lot of products have indicated to the farmer for the control of the disease. But at now, no product are authorized in Italy for the efficacy control of the bacterium, and also no the count of the bacterium is present from a long of time. No product were able to contain the disease in the field. So, in order to obtain some information regarding the effectively control of some product against the bacterium, from 2015 we have set up 4 field trials. All the trial was set up in the Lecce district in the infected area. In the trial E, we have used a 3 years old olive tree that have tested free from Xylella. The planted were put in the trial in a randomized block with 15 plants per each block. The plants were leaves to the natural infection of the vector during the first year, all the plants in 2 blocks, while the plants in the other blocks were caged, and in each cage were put a prefixed number of infected Phyllenus spumarius collected in an infected area in order to increasing the development of the disease. In this field trial, during all the trial that is 3 years, we haven't applied specific insecticide for the control of Phyllenus spumarius and others olive pests. Trial B were set up in commercial olive orchard with 12 years that showed few or medium or severe symptoms of the disease. The distribution of the infected plant at the beginning of the trial in the field is reported in this slide. As is evident, the few plants showed a symptom of the disease, but we have showed the plants that were arranged in each treatment in order to avoid the statistical difference about the treatment regarding the prevalence and the severity index. In trial C weren't symptoms of Xylella fastidiosa at the beginning of the trial, but the olive orchard where we have carried the experimentation were surrounded from ancient olive orchard with severe symptoms of the disease or dead plant. In this olive orchard, the farmer implemented the best agriculture practices with adequate control of the weeds, frequently pruning of the shoots both during the winter and during the season to remove the shoot dissected, and applied frequently insecticide to control the other pests of the olive tree. Trial D were set up in olive orchard without symptom of the disease and surrounded from olive that show few symptoms of the bacterium. The tested products. We have tested elicitor of plant defense, such as cerevisane, fosfetilalluminio, protein of arpin, cosoga, and cibenzolar S-methyl. Such of this product have used because have specific activity against other bacterium, and in particular protein of arpin have showed in other studies carried out in California activity against the Pierce disease in the reduction of the percentage of infected plant in respect and the control. All the products were applied in spray with interval application of 25 to 20 days, starting the application on the end of the winter when you shoots emergency until July, with the interruption in August and the 2 application in September and in October. Then we have tested N-acetilcysteine following the indication of Dottoressa de Souza, and we have applied N-acetilcysteine in endotherapy in 2 different rates at the concentration of 22 grams per liter and a double concentration. In one thesis, the product were applied alone. In the other thesis, were applied also to the soil. And then we have used as a control organic fertilizer, because in the case where the products were applied to the soil, we have used as a substrate for capture the NAC organic matter. The N-acetyl-cysteine was used in two trials, 3I and in trial B. Trial E, where we have planting a new olive plant, and we have tested the product at the rate of 1 gram per plant during the first 2 years of the activity, and 2 grams of plant in this year, with 50 milliliters per plant of the applied volume and 100 milliliters per plant in this year. And the double dose in the other cases where the product were applied at the highest rate. In this slide, it is evident the phase of endotherapy and then the application to the soil of the products. This product then was applied also in trial B in plants that have 12 years, and the rate of the product that we have applied is more high than 11 grams per plant at the lowest rate and 22 grams per plant at the highest rate. The product applied to the soil were applied at the dose of 4 grams per plant, and the organic fertilizer alone in mixture with the N-acetyl-cysteine at the rate of 4 kg per plant. This is the phase of the application of the products to the plant. So this is the phase of the application of the NAC to the soil. The NAC were absorbed on organic fertilizer because they release more slowly the product, and then were plugged in the soil. For the evaluation of the effectiveness of the tested product, we have used the empirical scale of 5 classes, 0 to 5. 0, plant is asymptomless. 1, a light symptom of the disease with at least 10 shoots with symptomatic. 2, light to moderate symptom of the disease with more than 10 shoots with symptoms, and 3, heavy symptom with at least 1 branch completely infected. 4, heavy symptom with 2 branch, at least 2 branched symptomatic, and 5, plant dead. In this slide can be observed the distribution of infected plant in the trial E, in which we have planted the new young plants. The square red is the infected plants. After 1 year from the starting of the trial, the percentage of the infected plant ranged from 20% in untreated check to 14% in tesi treated with acibenzolar S-methyl, without a statistical significant difference among the considered tesi. Then, after 2 years from the starting of the trial, the infected plant are all the plant reported in this slide. In tesi treated with cerevisane e acibenzolar S-methyl, 100% all the plants are infected, while the lowest value were observed in tesi treated with untreated check and NAC in endotherapy. At the same time, this is the actual situation, and in this study are reported the distribution of the infected plant in the field trial. prevalence ranged from 20% to more than 8%, without statistical significant difference among the considered theses. But it is evident that the thesis treated with the NAC showed the lowest value of prevalence, but we not expected the difference regarding the diffusion of the disease, but expected the difference regarding the severity of the disease. In fact, in this slide is evident that NAC applied in endotherapy have a statistically significant difference as other treatment, while the highest value were observed in untreated check, and also other considered theses not show the difference as the untreated. In this slide can be observed the plants when we have started the trial and the actual situation. Frequently we have observed infected plant when they are more vigorously, and it is evident the characteristic symptom of the disease. And in this image picture there are plants that show the severe symptom of the disease. In trial B, after 10 months after the starting of the application, the percentage of infected plant ranged from 75% in the thesis treated with NAC and endotherapy to 95% without difference among the different treatment. This is the situation of the infected plant in the field of trial after 1 year from the beginning of the trial. All the plants in each treatment was infected, with the exception of the thesis treated with NAC and endotherapy in association with NAC applied to the soil and adsorbed on organic fertilizer. Regarding the severity of the symptoms, it can be differentiated three groups. A first group in which are the more severe symptoms of the disease and include untreated check and the thesis in which we have applied as control the organic fertilizer. In this case, it is evident a peak of increase during frequent application of the product of the severity of the disease. Poi we have other group that included the treatment that have the lowest value of the severity of the disease, this acibenzolare S-methyl and NAC applied at two different rates at different mode of application. And then we have a medium value for the others resistance index that we have tested. In this slide can be observed the distribution of the class value in different treatment in the two different survey carried at the beginning of the trial and of the trial. At the beginning of the trial, it is evident that almost all treatment showed a value of severity equal to 1, with a few plants that showed a severe symptom with a value of severity equal to 2. At the end of the trial, the severity of the disease increasing notably in almost all cases, and reached a value equal to 3, that is plants with severe symptoms of the disease, with the exception the tesi, in which was applied H-benzolar S-methyl and NAC. In almost all tesi, then it is evident that the presence of a plant that showed a value of severity equal to 4. In this picture is evident the good vegetative condition of the one plant that treated with NAC and therapy in comparison with other cases in which is evident the completed dissection of the branches. So in trial C we have the same trend in that at the beginning the observation have observed the infected plant and symptomatic plant compare subsequently, and it is evident in irregular trend of the symptomatic plant, probably due to the pruning operation that carry out the farmer, and this is evident also regarding the severity of the symptom. In this slide is evident that in a few periods of the observation, phosphatyl aluminium showed good results, but these results are nullified then. This is the last trial, the trial D, in which are evident the distribution of the symptom after 1 year from the starting of the trial and after 2 years. The distribution of the symptomatic plant and the severity of the disease, but it is evident the equal trend in all the considered cases, without difference among the cases considered. In conclusion, the resistant inductor phosphatyl aluminium benzoate S-methyl occasionally showed best result, but it not always were confirmed. As others inductor, Kosoga, protein of ARP, and Cerevisian frequently showed symptoms more severe than untreated check. And also organic fertilizer. About the tested product, only NAC showed a promising result in both the trials where they have up if the percentage of infected plant were not reduced. Are ongoing other activity, but at now, no of the tested product can be indicated to the farmer for the containment and the remission of the disease symptom of Xylella fastidiosa. Thank you, sir, at all.
Thank you, Anza. And now Professor Franco Nigro from the University of Bari, mycologist specifically in fungal disease of olive, will present the endophytic bacterial community of healthy and Xylella-infected olive sapwood.
Okay, thank you. Good afternoon to everybody, and many thanks for this opportunity. When we're talking about the endophytes, there are some kind of definition that we must recall. Endophytes are usually organisms that colonize plant tissue without causing any visible symptoms, and are microorganisms that can be isolated from surface-infested plants or extracted from within the plants. And for sure, these endophytes do not cause any kind of symptoms. Sometimes we heard or find in literature that some microorganisms can switch from the endophytic phase to parasitic phase, but usually when you're talking about the bacterial endophytic community, this behavior is quite rare in literature. So if a bacterial species is reported as endophyte probably will not switch on the pathogenic phase. To the best of my knowledge, to date we have no report and literature indicating that some bacterial species behave in this way. There are a large evidence about the role that endophytes have in plants, both microorganisms, culturable on media or unculturable, are involved in industrial application, crop yield and plant growth promotion, and what is important for our interest now is plant health and protection, as well as recently some endophytes have been used as assisted phytoremediation to remove pollutants in the soil. And if we take a look to the bacterial species involved in this behavior, we can find some names, some genus that are almost the same: Pseudomonas, Bacillus, Pseudomonas, Bacillus, and Penibacillus, if we think to some antibiotics. Well, all this evidence have been obtained considering, herbaceous host. Very few indications are available about the role of endophytes in olive trees, and main of the data we have refers to the rhizosphere. There are several reports in literature indicating that several species of Pseudomonas are involved in the resistance to other systemic disease. Well, if the pictures, what the role of endophytes in the olive quack decline syndrome? At the beginning of this story, several species of fungi have been reported as associated to this disease, mainly related to Phaeacremonium and Pleurostomophora, and also some new species that have been identified, Pseudophaemoniella. And to date, based on the results we have got, we can say that the role of these fungi in the disease is almost marginal. And for those interested, there is a specific poster reporting the data of 2-year experimentation in confined environment about the role of these fungi in the disease. Xylella is a xylem inhabiting bacterium, so maybe we have some bacterium, some group of bacteria that colonize the same niche. And what is the role of this bacterium in the disease or in the development of the symptoms? So the objective of this new experimentation we started the last year was to characterize the bacterial endophytic population occurring into the xylem of healthy and Xylella-infected olive trees by an isolation-dependent approach. That is, we would like to have the bacterial colony just to be better studied and investigated. For sure, also the microbiome that are the mixture of culturable and unculturable bacteria may have a role in the disease expression symptoms, in the severity of the disease. And for those interested, there is a specific poster authored by Massimiliano and co-workers in the session number 3, the poster number 3.6, that can be viewed and discussed. So this is also important if we would like to draw some hypotheses about the behavior of resistant and susceptible cultivar. That is, may these bacteria, endophytic bacteria, have a role in the susceptibility or resistance of the olive cultivars? To this purpose, we considered the varieties that, as Donato said in his presentation, FS17 and Lecino are to date the most promising olive varieties as for resistant behavior. Instead, Kalamata can be considered one of the most susceptible, considering the data up to date available. So we consider in the open field some plants of this variety that were infected by Xylella, and those that still resulted health in the field. This test was done by the PCR analysis by Dr. Saldarelli and Massimiliano. To detect the endophytic bacterial population, we use lignified shoots taken from the plants that were subjected to to treatment. The bark was removed, and the shoots were sterilized in sodium hypochlorite, then in ethanol 70%, then the xylem was minced and suspended in buffer, and then the suspension was seeded on a different medium from PDA to BCYE after appropriate dilution. And all the bacterial colony that arise from this cultivation medium were identified by sequencing the 16S region of the rDNA. So we have some preliminary data. To be honest, we are working with the material from the field, and the results show very, very high variability. However, some trend can be individuated, at least if we consider these preliminary results. That is, if as a general rule, in the healthy plant, apart from the variety, we have a little bit higher population of bacteria as compared to the infected one. Also, the other microorganisms have been counted, yeast and Basidium, that are the common endophytic microorganisms in olive. But as we can see, there is no clear relationship among variety and the condition, healthy or infected. If we consider the other data available in July, as general data, we can see that the endophytic population tend to diminish. We started from 4 units of log, and here we have about 1.5, no more. So maybe the temperature have this effect, the high temperature have this effect to reduce the bacterial population inside. And the trend appeared the same, even if in this case it seems a little bit much more pronounced, the difference between the healthy and the infected, being the bacterial population higher in the healthy plants, in non-infected plants, as compared to the infected one. Among the different species that, genus that we have identified, we have Bacillus, Pseudomonas, Panteo, and so on. With some species very interesting as the role they may have as biocontrol agents, such as Rhodococcus, Sphingomonas, and Stenotrophomonas. Particularly, our interest focused on pigmented facultative methyllotrophic bacteria, a particular group of alpha proteobacteria in which Methylobacterium is the most common, and also is the same genus that has been reported by Brazilian researcher as involved in a mild expression of symptoms on citrus affected by CVC. And also, if we consider the footprint of the squeezed shoots, we can see that this colony is quite common. So this means that Methylobacterium occupy the same niche of Xylella fastidiosa pauca into the olive xylem. And the literature, the data in literature demonstrate that the role of Methylobacterium depends on the species we are considering. Methylobacterium estorquens is reported in the citrus plants that show high, severity of citrus variegated chlorosis. Instead, Methylobacterium mesophilicum is associated with healthy citrus plants. These are the data in literature. So, moreover, in vitro studies demonstrated that Methylobacterium mesophilicum could reduce the growth of Xylella in vitro. Instead, conversely, Methylobacterium estorquens had no effect on the growth of Xylella. So the authors did the hypothesis that the development of citrus variegated chlorosis depends on the equilibrium between these two species. As we, during the trial, see that on the BCYE, the number of these red and pink pigmented colony was quite easy to be counted. We did this observation comparing the resistant with the susceptible variety, but infected or non-infected, and we can see that the population show a trend. There is no significant behavior, absolutely. We have a huge variability of the data. Also, if we use 8 plates for each samples and at least 5 plants for each combination, variety and health condition. So we can see that we have this trend, a tendency to high number of red pigmented colonies in infected plants as compared to the healthy plants. The same in the next assessment, July 2017, on the CYE. The trend was the same, but we just saw a higher difference as for the susceptible Kalamata that was infected with Xylella. So, do these have a meaning or an effect on the resistant susceptible behavior? We don't know. This is just preliminary. This is just the first phase. We need to do some more investigation. So the problem now is, what about the species of Methylobacterium when found in olive? We've been identifying 2 or 3 isolates, the most frequent. By using the 16S sequence, we have 99 overlap with the Mycobacterium mesophilicum, but just 2 base pair difference, and 4 base difference with Methylobacterium brachiatum, and 7 base pair difference with the Mycobacterium pseudosacicola. So, to date, it is not possible to assign our strain, our isolate to a specific, to a species, because the difference appear to be relevant for the identification. Much more interesting is the isolate GR19, which have 98 overlap with the available sequence. And so we don't know if we have new species or a particular strain. For sure, also if we consider some ability and use some carbon source, the 2 isolates we characterize, GR19 and GR22, have some different behavior as the codified characteristics for the species Extorquens and Mesophilicum. So the identification of this strain is quite difficult. We are now trying to amplify and sequence the GRB region, because it's reported literature as the most important region for a precise identification. So the role of this Methylobacterium in olive, maybe is important because it's involved in the carbon cycling. So maybe this is related with the production of exopolysaccharides, so biofilm formation, who knows. It has been reported an important role as a modulator of the response to abiotic stress because Methylobacterium are related to the production of some hormone product in plants, and also mainly for the rice, there is the ability to fix the atmospheric nitrogen. So what is the role in olive? We don't know. So as concluding remarks, under fit condition, the population level of cultivable endophytic bacteria was highly variable, being affected by the host, genotype, and maybe wilting severity. A trend indicates a higher bacterial population level in the health than in the Xylella-infected plants, and it seems that Methylobacterium have an opposite trend as compared to the other cultivable bacteria. So the identification of the Methylobacterium involved in the olive, or the most frequent species, could be important to give an help to foresee if these are useful as a potential biological control agent or not, or just a scientific speculation. Thank you.
Thank you, Franco. The next presentation is by Massimiliano Morelli, a researcher at the Institute for Sustainable Plant Protection, CNR Bari. His presentation is on attempts to develop sustainable biocontrol strategies of Xylella fastidiosa infection in olive.
Thanks, Maria, and thanks to everybody and to EFSA for funding the participation of young researchers to this event. Here we report our preliminary attempt to develop a sustainable biocontrol strategy for Xylella fastidiosa in olive. Our effort traced their origins back to some keystone studies that were started from the Berkeley group led by Professor Lindo, and that clearly defined for Xylella fastidiosa a dual lifestyle in which two different phenotypic behaviors coexist. And as you can see, this switch is mediated by a series of features that are, of course, following different patterns. And behind all this machinery, there is an important aspect, a cell density dependent signal that was defined as DSF, as diffusible signal factor, that seems to drive all this changement. As we know, this DSF factors have been chemically characterized, and at least two different compounds has been proved to be biologically active on Xylella fastidiosa. And this is a common feature shared with other gram-negative bacteria members. Behind this scenario, there is a complex and fine-tuned regulated gene cluster that has been defined as the RPF gene cluster. Roughly, we can summarize at least three main components of this system that are an RPFC sensor, an RPFG regulator that is able to up and down regulate a cascade of genes involved in this change, and the last but not the least, the RPFF gene. This is a synthase that is responsible for synthesis of DSF factor, and as you will see, will play a main role in our story. What we want to do, we are trying to walk through a sustainable strategy that is capable of pathogen confusion. To do so, we have to follow a preliminary step, that is to know what's happening in the ST 53 strain causing olive disease in Apulia, and what is the wild-type behavior and in a relationship with the DSF produced by this strain. Moving on, we want to develop a pilot study in which we can handle a model system, trying to understand before on indicator herbaceous plant if an alteration of the natural behavior of regulation of DSF can lead to, hopefully, to a decreased virulence in plants and in some way to limit at least the infection around the point of inoculation. To start, we look at an heterologous system. We tried to include the RPFF gene taken from our strain of Xylella fastidiosa into an expression system to produce the RPFF encoded protein. The same we did for a control gene, a BFP gene. What we saw from the beginning in SDS page analysis and then confirmed by Western blot, it was that we were able to produce huge amount of the two proteins. So we give this first crude extract to our chemical staff, and they try to have a first sight of what's happening by gas chromatographic spectrometry analysis. What they see is that from the retention time analysis and as confirmed by the fragmentation pattern in mass spectrometry, it seems that the E. coli harboring RPFF gene is producing a series of molecules that can be putatively associated to fatty acids with a chain length comprised between 12 and 18 carbon atoms, so according to the expectation. Of course, they are working now to get a further purification of this complex matrix, but in the meanwhile, we wanted to have a first measure of a possible action of this crude extract on our wild-type strain. So we had to make use of a widely used method for a preliminary assessment of biofilm formation. And as you know, the Xylella cells are capable of creating a ring of attached cells to the surface of cell culture tubes. And then, so when we draw off the floating cells, the planktonic cells, you can you can stain with crystal violet solution, and then after washing steps, dissolve into ethanol and measure the OD 600 nanometers absorbance and have a measure of this biofilm formation quantity. So what we saw, it was that when we applied a serial dilution of the two crude extracts from RPFF harboring E. coli and those harboring the BFP control gene, we saw that it seems that there is already an action of these compounds on the biofilm formation when compared to the control. And this is true at 24 hours and also at 48 hours. And with the main effect with higher dilutions. When we test these things, we should also take into account that when you want to measure an exogenous DSF effect, you have also to consider that there will be an endogenous DSF effect. And so you should choose the right time where to have this measure, because, of course, when the wild-type effect will start, it can cover any effect of exogenous application. So we also are proceeding with some characterization, because there were no data available, of course, on our strain. And what we saw is that there is a major shift between the 24 hours and the 48 hours. So this can be a key point in our measure. And I can add also that, as we mentioned several times today, there are a series of genes that are involved in this change. And so we have a lot of genes that can be studied, panel of genes that can be studied as a differential expression at different time points. Here I can show you one of these analyses. You see, you see that one of these genes, those encoding for hemagglutinin-like protein B, has also an overexpression at 24 hours, and then it starts to decrease. So this frame shift between 24 and 48 hours can be of relevant interest for our analysis. But to do what? We wanted to go back to our established expertise as plant virologists. So we try to create a vector based on tobacco mosaic virus that, that is harboring the RPFF gene from Xylella fastidiosa. To what aim? To try to exploit the natural and established high replicative attitude of TMV virus to deliver, to faster deliver the RPFF product into the plants. In the same plants, some indicator plants, we can also co-inoculate Xylella fastidiosa and see if there is an effect on this alteration of natural DSF regulation. So we started by constructing this vector. What we did it was to amplify the Xylella DNA flanked by 2 restriction sites to clone this into a commercial vector to have an high copy number. And then we use an already available vector where it was included a GFP gene. We substituted the GFP gene, and we then included our Xylella gene, and then we have a full our vector where the exogenous gene was posed under the control of a dual promoter. We proceed with the transcription of this vector into a fully infective clone that was inoculated to Nicotiana benthamiana plants. What's happened? We saw in Western blot analysis that while at the first round of inoculation, the protein was clearly expressed and systemically delivered into the plant tissues. After the second and then the third round of inoculation by mechanical transmission, it seems that this protein starts to disappear, or at least it reached an undetectable threshold. In the meanwhile, the virus is still there and continues to spread into the plants, shows clearly symptoms, and at the same time, we also use the original vector harboring the GFP protein, and you can see that also in that case, the protein is dull and is capable of showing bright existence, as you see in the picture. So what? Now we have to calibrate this system to see what's wrong there and trying to make better this system. But in the meanwhile, we are already starting an alternative strategy. In this case, we are making use of an agrobacterium-mediated system in which we are inserting our previously realized vector into a binary vector, and in this case posed under the control of cauliflower mosaic virus promoter. And with this system, we will exploit the established ability of agrobacterium to integrate a tDNA containing our gene into the genome of the plant, and then in this system we will merge the effect of agrobacterium, the efficiency of agrobacterium in reaching a stable expression, and the already mentioned ability of TMV to deliver systemically the protein of our interest. That's all for now, but we are performing a lot of work, and we are confident that in the next months and in the framework in the framework of the two projects funding these activities, we will come with interesting results on these premises. First, to close my presentation, I want to thank the funders and mainly people. Firstly, Juicy, as she's behind my words. There is especially her hard work during her PhD thesis, and our collaborators and our mentors, Maria and Pasquale, and finally Professor De La Fuente and Professor Alessandra de Souza, as they are always promptly answering to our queries, and I'm sure that their suggestions are and will be more than welcome. Thanks to the audience for the attention.
Thank you, Massimiliano. And now we move to the last presentation, which will be given by Professor Emilio Montesinos, University of Girona. And he will give a presentation on antibacterial and plant defense elicitor peptides against Xylella fastidiosa.
Thank you, Maria. I would like also to thank to the EFSA and the organizing institutions for accepting my contribution to this important meeting. As it was clear from the previous presentations, from now and this morning, there is a need of novel compounds to fight against Xylella fastidiosa. If you look at in detail, you have seen that practically none of the compounds that have been tested in the field are new. So they are all compounds, even including the N-acetylcysteine that comes from the pharmaceutical field. So our approach was that first to find compounds from nature that can be important in the control of Xylella and other bacterial diseases. So one first approach is the field of antibacterial and antimicrobial peptides, which are polypeptides of less than 50 amino acids. This is for convention. The source of those peptides is in the world of animals and plants that are the first barrier of the immune defense on the plants and also in many animals. I'm not going in detail to this type of compounds, but also in microorganisms, and play a role in several microorganisms in the antibiosis processes for competition for resources in the nature. We now know more than 1,000 compounds of different types. So in my laboratory, 15 years ago, we started a collaboration with a peptide group, peptide research group, which is the LIPSO. My lab is the CITSA. And these are expertise people that synthesize and design peptides on the basis of natural structures and on the basis of design function approach. So departing from natural compounds, we started a round of molecular design, the synthesis using combinatorial chemistry of different families of peptides, then testing and screening for antimicrobial activity. Now we have included Xylella fastidiosa subspecies and a few strains. And then in a first round, we have lead peptides, and these lead peptides can be improved in a second round with a new design, new synthesis, and after 2 or 3 rounds, we get with real collection of lead peptides. But these lead peptides have passed also a process of analysis of hemolytic activity because we don't want to have toxic compounds. So this is a test which is very easily done. And also to protease stability to prevent that the products are rapidly degraded in nature, especially within the plants. So these improved peptides gave us mainly 6 peptide libraries. These are the structures, but the ones I will go to talk today are the linear secropin-melittin hybrid peptides, which are andecapeptides made on 11 amino acids from proteinogenous origin. So there is no amino acid strange to the system. But we have also cyclic peptides, biaryl peptides, peptidotriazoles, carbopeptides, and also cyclic linear lipopeptide. So this is a whole family of compounds that can be tested against any pathogen. So the first candidate to be applied in the Xylella fastidiosa control was this BP100 peptide, which is a very simple, just 11 amino acids with this sequence. This is a one-letter sequence. You can see many Ks here, and this is because there are lysines. So this peptide is a cationic amphipatic peptide. So the structure is something like an helical wheel. This peptide has been tested even into the field against several diseases. I'm not going to detail, include fire blight, which is caused by Erwinia millibora, several Xanthomonas diseases, even fungal diseases. So we have analyzed it in detail, this peptide. There's a second candidate for the project, which is this BPC-194, which is a cyclic peptide. You can see here the structure. And this is the three-dimensional structure. So like the previous peptide, BP100, these peptides are binding to the membrane and making pores in the membrane of bacteria. So they are killing the bacteria in a very, very beautiful way. So one important thing is that the minimum inhibitory concentration is very low, just in the range of several antibiotics. So these two compounds are obviously patented, and they are under development. And the last peptide candidate is this BP178, which is a derivative from BP100 that was designed to be expressed on plants. So if you are expertise on that, you will see this sequence and this sequence, which are special sequences to favor the expression without killing the plant cell. And this is the structure. So essentially, the minimum inhibitory concentration is similar, but one very important aspect is that this peptide has an activity as plant defense elicitor. So this is, for us, one of the last generations of peptides. Just very briefly, this is a list of genes that are involved in plant defense or plant stress relief. This is the case of tomato, in which we have been working several years. And you can see here that BP178 peptide gives this red, means overexpression of these genes after topical treatment, which is very important. And you can see here flagellin 15, which is a microbial-associated plant defense elicitor as a control. But you have ethylene, jasmonate, salicylic acid also as reference. And you can see here that this peptide is really stimulating the expression of genes involved in defense. For comparison, if you go to BP100, you see that is only in 2 cases doing this effect. So BP100 is known effect on plant defense elicitation. So we don't know what happens with olive trees, with prunus, with all these hosts for Xylella, but we expect that this is working in a similar way. Then another source of peptides for us in our research field is the cyclolipopeptides that came, for example, from bacteria like Bacillus or Streptomyces or Pseudomonas. So one of our projects departed from Bacillus amyloliquefaciens strains, and this was developed for a company and also in a national funded project. But you can see here 4 of these cyclolipopeptides. We extract the peptides from cultures, and by means of mass spectrometry analysis, we identify iturines, phengicines, surfactants. Interestingly, the surfactants are a kind of biofilm inhibitors because they really have a kind of effect, is a detergent activity. But iturines and phengicine have been not studied against Xylella fastidiosa. So these sources of compounds were used, and the tests were made by Isabel Mora, which is here in the audience, in the front back. And we did this test under a very, very strict and difficult conditions. And I was surprised that nobody that gave presentation previously talk about that. So in Spain, we cannot work with living Xylella fastidiosa unless you have a very strict containment facility. So as far as I know, in Spain there are two places. One is the laboratory from IBIA, that there are people here working. Another one is my lab. And now we are restricted to a very small lab which fulfill the conditions for Xylella. But curiously, these conditions are more strict than for the common A2 quarantine bacteria. So we know that Xylella is, at least as far as we know, is not transmitted by shoes, by water, and things like that. But this is a condition. So the laboratory should have containment apart from the normal ones. You cannot go out with contaminated plant material, of course, but also you have to prevent vectors in the lab. But the other conditions are like level 3 containment. So this is very strict in my view. But in any case, we have a greenhouse which fulfills the A2 level, and this is authorized, but not for Xylella. So can you imagine the experiments in this small laboratory? Okay? So the experiment was very simple. We work with the living Xylella, so we are very worried about to have escaping cells from that. So the test was a very easy test, which is the killing or contact test. We have the suspension of the bacteria. You have here the pure culture suspension in contact with the peptide or the product from natural origin. There is a contact test made on phosphate succinate buffer, and then by serial dilutions, we spread onto the plates, and we determine the survival. So this is a culturable method to evaluate survival of this bacteria. We tested the main 3 media for growing Xylella, and the best performance was on PED2, and also with this phosphate succinate medium. So here there are the results. Up to now, we have only 3 strains. We have the Temecula, one subspecies fastidiosa, and we have also the Cordeiropolis strains that were kindly supplied by Maria Saponari, the Cordeiropolis strain, and Temecula strain was supplied by Esther Marco from IBIA. And this is a collection strain. So you know that since you are importing a quarantine pathogen, you need permits, so there's a lot of bureaucracy and administrative requirements. But once we got the strains, we did the experiments, and this is very interesting because peptide BP178 had a minimum inhibitory concentration or a median killing dose, which was in the range of a strong activity. But interestingly, you can see that Cordeiropolis strain is less susceptible than the other strains. And you can see also that there is a dose effect. For example, you see here 1 micromolar, 10 micromolar, and 50 micromolar. So there is a practically linear relationship. So the products are active against Xylella. We don't know what will happen with BP178 once we apply to olive trees if there is an induction of defenses. But now we have a good candidate to proceed in the next studies. So the other source of cyclolipopeptides was a strain of Bacillus amyloliquefaciens that produced fungicins, surfactants, and iturins, and now we have also different bacteriocins, for example, basilicin, and there is a large amount of different peptides available. But this is also, again, interesting. So this is percentage of inhibition against the 3 strains of Xylella, and we find that iturins and fungicins were very active, but surprisingly, the surfactants were practically non-active. So in the future, we will concentrate in these iturins and fungicins that we can obtain in pure state to make the tests. So the question after that is how we deliver these peptides to the plants. So one alternative that we have seen in the previous talks is endotherapy. So people say, wow, are you going to inject the peptides to 1,000 years olive trees? The answer is no. This is not our idea. Our idea is to do this approach, for example, at nurseries, at places where you can protect the trees from the problem, from infections, or even in the in vitro stage, for example, in nurseries. So that's the idea. But for doing some previous tests, we have been testing these peptides in a H2020 project, which is the DROPSA project, deal with Pseudomonas syringae actinidia in kiwifruit, with Xanthomonas pruni in different prunus, and also with other pathogens. But we have tested a system which is rather different from the one used by our previous colleagues, which is a machine that is really an automatic machine that you can use to inject even trees. So this is used, for example, against the red cork in palm trees to injecting insecticides. By the company, which is Endotherapy, which is a Spanish company, has developed a prototype machine that can inject even 1 milliliter of peptide. So this is good for us to make the test. The question is that BP100 peptide injected is active. So is active against this bacteria. We don't know what happens yet with Xylella fastidiosa, but we expect similar results. So the added value is that BP178, which is a plant defense elicitor, would require considerably lower concentrations than with bactericidal compounds, because you are eliciting defenses on the plant. So this is merely now an hypothesis. The next stage in the XF Actors project will be first to test toxicity of these peptides on the Xylella fastidiosa hosts. We expect that no problem because we have tested previously in other plants. So that could be, but it's very rare. And the second thing is to use a model plant. So for us, there are 2 model plants up to now, or 3. One is tobacco, which is a surprise, which developed the disease symptoms relatively fast. The other is olive trees, olive plants, microplants in our case, but also prunus. And we are in contact with a company from Spain that is very popular here also no, not here, in Italy, not in the islands. And they work with in vitro prunus rustox. So these plantlets are very good for our purpose. So the future will be that. Maybe in the next meetings, I will show you the success or unsuccess of this approach that I think is novel. So these are the conclusions. Essentially, the field of antimicrobial or functional peptides is very open. You can tailor peptides for any target you want. This is my message. So if you know a process in which there is an interaction, for example, you can interfere with this interaction if there are proteins or peptides involved. So an idea is to develop peptides to target the insect vector, to prevent acquirement of the pathogen. So this is one idea. The other is that these chimeras of peptides that have double activity, which is bactericidal on one hand and plant defense elicitation in another hand, are very promising because we use 2 targets in the process of interaction of the pathogen with the plant. Cyclolipopeptides from natural origin, so this is for those of you that are thinking in organic agriculture, coming from microbes can be also future compounds. Of course, in the case of Xylella fastidiosa. But don't forget one thing, because I have seen about acetylcysteine. So if you try to register this compound in Europe, you will get problems, probably, because it's a clinical product. So in our case, if a company is interested in such compounds, we'll need registration. So registration means a dossier, a very long dossier, but don't forget, peptides, in our case, were designed taking into account biodegradation, low toxicity, and several aspects that could probably fulfill a requirement for the approval. But there is one problem that is not still solved, is the production of the peptide at high level. So could you imagine how is the cost of 1 gram of peptide up to now? Is about 1,000 euros per gram. So we have only a dose for a big tree. Okay, so we need to improve synthesis, to improve processes. Up to now, we have produced BP178 in rice seeds as a biofactory, because this peptide was designed for expression on plants. But now we are trying to produce that in a microbial factory. So we expect to decrease the cost of the peptide to levels that can be compatible with agriculture. So this is the message. I would like to end my presentation with a contribution. There are several researchers from the University of Girona that have contributed in the past and also now. Also from a center, which is the Center for Genomic Research in Barcelona, and the funding from the Spanish government and also from the Catalonia government, and different projects from the European Commission, CEPSAPE project, which was an ARNet for production of peptides in plant biofactories. The DROPSA is 2020, which is a recent, the last FP7 call, and now XF actors in which we participate as an in-kind contribution with the CSIC from Spain, which is led this work package by Blanca Landa, which is here. And finally, I would like to thank to all of you for giving me the opportunity to talk about this field of peptides. Thank you very much.
Thank you, Emilio. Emilio, and we have 15 minutes for question. So, she's arriving.
So a question about the use of peptide, because to Emilio, I'm not sure whether I thought about this product could be useful in agriculture. I'm not sure about the nursery, because my idea about nursery control is more what is done in Brazil for the CVC. So it's screening and treatment to avoid that the insect vector come into the nursery. So I don't see the reason why to treat endotherapeutic a plant for something which should not happen, because you have a prevention from the transmission.
Yeah, thank you, Giuseppe, for the question. You are right, but as I always said in the past meetings of Xylella, in my laboratory we are thinking about what to do with diseased trees, other than eradication and burning the trees. This is the question, and I think this is an important question. So our idea is to try obviously not to apply as a preventative strategy to all the trees, but for example, in the case of an outbreak in an area, maybe to complement such method of peptides with heavy pruning, nutritional immunity, as this morning has been talked about that. Let us say a combination of systems. I don't think that peptides by itself will have a solution, but well, in any case, is a novel field to be developed. So in the future, our plans are to develop peptides to trigger the vector, and especially, as I told, the vector acquirement of the bacteria. And we have plenty of ideas. The only thing we need is money to develop such ideas. As you know, probably everybody here is thinking about the same, but this is an important question. So peptides can be designed, and as you know, in the same way that interference RNA now is really a field that thinks to solve everything, we thought that peptides could contribute. This is not new. Our peptides are new, of course, but this has been, Alexander Pustel knows very well, have been done in the United States against Xylella fastidiosa, but the approach was not this approach, was to express the peptide on the plant. So in Europe, if you get a GMO plant, you are lost. So our idea is go to the standard way, which is to develop products that can be accepted by the administration. But this is, thanks.
Other question? Martin, yes.
Emilio, to go further on that topic.
Oh, yeah.
But don't you expect a rather transient action, a temporary action of these products? First, what is the lifetime? Secondly, when you have still an active plant system, in the xylem, it is drained away because the xylem and it is expelled from the system. Yeah. So then it would rather be a continuous providing of the product, or rather big time to provide it.
Yeah, Martin, it's clear that there is nothing completely clean. So even if you apply peptides into the plant, you are probably shifting the microbiota diversity on the plant. You are doing changes on the plant that we don't know. We have to examine that. But one thing we have observed, for example, with P178, is that plants tend to be a little bit bigger, so more vigorous. We don't know why. But with other peptides that elicit plant defenses, we have observed really a stop of growing. So they act like hormones, like phytohormones. So this is the reason for what we think we have to test much more the peptides, especially in the field, because probably the shelf life of some of the peptides could be very short, but for others could be longer. But this can be designed. So it's a question of introduce an amino acid in part of the sequence that is hydrolyzed by a protease. So it's simple like that.
Yeah.
A couple of questions to Emilio. One is for, related to my ignorance. How do you detect the peptides, if it's possible, in the plants when you have injected this, after you have injected the peptides? So there is a means, a tools to detect the peptides in the plants. And the second question is, how do you explain the lack of effects on the donor strain, on the ST, because of the accessibility to the bacteria cell wall or biofilm or something like that?
Yeah, I'm going to answer the second first, which is, well, it's not a lack of effect, it's a less effect, because 50 micromolar is still a reasonable concentration. You have to realize that these are non-formulated peptides. So these are just diluted in distilled water and applied. So formulation generally increases the performance. So this is one thing. So what is clear is there is a differential response between subspecies. Well, between strains, because we don't know what happens with more strains within the subspecies. We are going to test that. But this means that probably one thing could be the outer membrane and outer structures, because such peptides have to make pores in the membrane as a first mechanism. So anything that can block, for example, if the bacteria has an exomucopolysaccharide layer, the peptide is cationic and tends to be binding there and do not access. So this is a kind of molecular engineering that should be done. And the other question, Oh, yeah. Yeah, this is very, well, it's very complicated if you don't add attack. And generally, what we do is a fluorescent tagging, but don't forget that if you add anything to the peptide, you change the peptide spectrum and activity. So you have to test this fluorescent tag to be sure that this is not the effect of the fluorescent tag you are seeing. Okay? Thanks.
So I have a question for Emilio Montesinos and then a more general comments also for Crescenza Dongiovanni. Yeah. First of all, I would like to know how can you figure out to deliver antimicrobial peptides to insect vectors? Okay. And then the consideration about these, in general, products, antimicrobial peptides, but also resistance elicitors, inducing. When dealing with resistance elicitors, maybe the application of such elicitors to plants that are already heavily infected, maybe it's too late. For example, in the case of Flavescens doré, okay, I'm a bit skeptical in comparing these two systems, but anyway, for example, the Bion product, acibenzolar S-methyl, proved to have some activity in preventing transmission of plants that were elicited before being exposed to infectious vectors. We found that it was completely unuseful in curing the plants. So what do you think about the perspective to test resistance elicitors before inoculation plants?
Yeah, well, the first question is about how to deliver to the insects. There are a few tests, I think one is published, not against insect vectors, but against an insect, which is Drosophila suzuki, by a group which is a partner in the DROPSA project with peptides that are interfering with the metamorphosis of the insect. And this is delivered in insect traps with feed. So simply is mixed with something that the insect is feeding. I don't know this insect what is or what can. Yeah. So yeah. So that's the one problem. The other is to use that as a common insecticide. But the thing important from the ecological point of view is that probably you will not kill the insect. You only will prevent the interaction with the bacteria. So we don't know what happens with this bacteria and the insect. There are people here like Alberto Ferreres and others. I think they know very well this kind of interactions, but we don't know still if the insect is suffering a disease or really is not suffering anything. So we'll see that. So this is one addressed to the target insect, and the other is about there is with all the elicitors there is a very short window in the phenological state of a plant that your elicitor is acting. So this is for bion, for phosphatyl aluminum, for any kind of elicitor. The problem is that this changes between the species and even sometimes cultivar. So in our experience with pear and apple, if you don't fulfill this window, you don't have any effect. So that could be another problem. But in any case, we are doing research anew on that field just to find which is the best time to apply elicitors to the plant.
Okay, and so do you want to add something?
In our trial, we have tested the resistance inductor before the on healthy plant in Xylella A, and before the infection of the vector, and in two trial on plants without symptomatic plant. Only on one trial the plants are asymptomatic. In any case, the behavior of the tested product is the same. Only acibenzolare S-methyl and phosphatidylate showed an effect only in one trial, and in the other trial we haven't the effect, with more application that we have carried out for the season and also for 3 years for two trials.
Yes, I have a question to Franco. Do you have any information how it's possible to inoculate interesting endophytic bacteria inside the plant, how to manage the population of good candidates you can select to have an effect of the endophytic bacteria against the target one?
Well, apart from the classic approach by endotherapy, that in the specific case of very aged olive plants in which you would like to save the crown and the big stem of the plants, maybe the endotherapy could be an opportunity if it works. But in literature are reported many cases of inoculation, if we can use these terms, of endophytes by spraying during, let's say, the flowering. Or there are several yeasts, such as Cryptococcus and so on, that are able to penetrate inside the tissue by using the stomatal openings. So the problem is, what is the role of these endophytes? Because the application of external endophytes during the flowering is common practice. It has been reported for strawberry, but also for olive, in which several bacterial isolates and also yeast have been applied during the flowering, just to see if the number of these endophytes increase in the drupe. So there are some data demonstrating this effect. The problem is, what is the role of these two species in the disease?
We have time for maybe the last question. Okay. Here.
I think I'm sitting in the wrong place. I always seem to be missed, but thank you anyway. Another question in the same vein from the last one, to Franco Nigro. I really appreciated your presentation, and I was looking at some of the results and thinking about the non-bacterial stuff that you were getting out. And you talked about Oreobasidium and various yeasts and stuff. But I was a bit curious.
What species?
The Oreobasidium
yeah.
and the other yeasts that you had, etc. I was just wondering, where are the filamentous fungi? Are there other fungal species?
I have data, yeah.
And is there anything which is showing any type of promise that has any type of antibacterial action against Xylella or anything in that capacity? I just thought it was interesting that I didn't see that profile. And you may have that data, and it'd be interesting to hear about it.
Yeah. Of course, we have also data on the fungal population that act as endophytes. And I presented some results in a previous research project meeting, the PONT project. And, well, there are several fungi that use antibiotics, antibacterial compounds. What I've seen is that in the infected plants, the number and the variability of the species that habit into the xylem is higher. And it is not easy to discriminate among the different species. We have a list of fungal species. We have not reached the terminal part of the work, that is genus. We have not data about the species. I have some data about Phaeacremonium and other fungal species that at the beginning well believed involved into the disease. Yes, it is another phase of the medal, but in my opinion, the movement of a fungi into the xylem could be a little bit complicated due to the dimension of the pit, and so I can figure out trichoderma that is able to colonize the xylem and produce, I don't know, maybe it is a fact of dimension. So for a bacterial cell to be much more easy to move inside the xylem, but for the hyphae or just conidia, it will be much more difficult to move inside and reach the terminal part where the infection starts. And another problem is that if we consider the possibility to spread an endophyte, well, the passage into the vector could be much more difficult. Instead, with a bacterial species, it could be much more simple to spread through the vectors.
Thank you. The last question, Leonardo de la Fuente.
It'll be short for Alessandra. For your work with NAC, two short questions is, one, I don't know if you mentioned how you look into what is the effect on the population of the bacteria inside the plant when you add the NAC. Another one, if I understand, so your hypothesis is taken by the roots and then breaking the biofilm in the xylem. Have you tested other, or are you planning to test other bacterial pathogens that also live in the xylem and form biofilm?
Okay, Liu, thanks for the question. And the first one is related to the population of the bacteria inside the plant, how I tested. We analyzed the bacteria, all the plant was already infected, and we analyzed the population before the NAC application and 3 months after NAC application by real-time PCR to analyze the population. That was the methodology we used. And the other one is to another bacteria, the answer is yes. We have the Xanthomonas citri, that is a problem in Brazil to citrus because it causes citrus canker. And actually it's easier because this bacteria form biofilm on the leaves, and so you can spray NAC. And so we published a paper last year that showing that NAC could be used to Xanthomonas citri too. And in vitro, it was in plant. And in vitro, you tested for other bacteria like Pseudomonas and Xanthomonas, and it worked very well. The only one that was not so good was Erwinia, that is a pectal bacteria, so it's a lot of the concentration of NAC to see the effect. So I hope that I have. So I'd just like to complement one point that was talked by Dr. Yes, sorry. And so because the regulation, we are working on this regulation in Brazil, and actually in Brazil it's very hard to regulate products to be applied in the field. And because this is a small company that was opening, we started to see the way to regulate NAC in the field, and actually it's not so difficult because all the tests in human was already done by the pharmacy, so it's not tox for the human or babies, it's not antibiotic, it's only a cysteine analog of an amino acid, so it was good for us. And so the indication is to regulate to like a fertilizer first, and then as a molecule that can kill bacteria. So this is more difficult, but if you start to regulate the NAC application as a fertilizer, it could be used because we proved it already that it has antioxidant effect. And so we already tested in the environment, and you see that it is degraded in the environment, so there is no residue, especially when we compare with the other molecule that's already used in the field, like copper, for instance, that is a heavy metal and stay in the soil, and it's used in the agriculture. And so we think that we have a hope in the future to transform it in a product.
Okay, thank you to all the speakers and for your attention. I think we can close this first day, and Miguel, if you have to give some information.
Thank you.
Sorry for cutting the discussion, but we really need to move for the poster session and networking reception. That is going to be 6:45. That is not already 6:45 to 9. But please remind you that presenters for poster session 1, 2, 3, 4, and 7 are requested to attend the poster from 7:15 to 8:30. And then also to remind you that from 8 to 9 in this same room, we have the special parallel session of Cure XF. Okay? Thank you.
Alcohol.